And get started. Thank you again, everyone, for joining us today for the Knowledge Portal Focus Group for the March meeting. We're just going to kick it off with some news items about Knowledge Portal Network, and then we will hand it over to our featured speaker. The first that I want to bring everyone's attention to is the American Diabetes Association Scientific Sessions, which will be June 5th through 8th in New Orleans. We are happy to announce that we will be a part of the NIDDK Scientific Corner. We are booth 2324, and we are very excited. Please feel free to stop by if you're going to be at ADA. We are extremely excited for another year at this conference. We will have, a lot of different basic if you have training sessions, if you have questions, we'll be promoting our new production release, which will be happening in conjunction with this as well. We're really looking forward to it and hope to see you all there. Another series that we have that is another webinar is through the Common Fund Data Ecosystem, which is another funding mechanism for our organization. Our next session for this will be Friday, March 27th from 1 to 2 p.m. Eastern, and we'll have some updates from the Knowledge Center as we will have our production release for this as well. So please feel free to join this if you are interested in learning a little bit more about another resource that we work on. And then one programming note, we're going to be moving to bi-weekly webinars. So this will be the second and fourth Thursdays of the calendar months. I'm going to be sending a calendar invite that will be updated after this focus group. This is geared towards, having more scientific talks, having more community members being able to participate in these events and such. So we're really looking forward to seeing you more frequently. As always, these will be recorded and they will be shared after the sessions. And we're really looking forward to this, and hopefully we'll see you guys there. So with all of that, those are my major scientific updates from the Knowledge Portal Network. And we're extremely excited to have one of our colleagues from UCSD today who will be showcasing his work on the single cell mappings. So with that, I will hand it over to Luca.
Hello, everyone. I'm happy to be here. I am starting to share my screen. Let's see. Can you guys see my screen?
Yes,
Great.
we can.
Sorry, I realized I was on mute.
It's all good. Okay. Hello, everyone. I'm Luca Ducciarone. I'm a senior postdoc in Cal Galton's team, and I am the main person leading this effort of creating single-cell maps for metabolic-related tissues. I led this effort, but the main person that actually worked on the dataset is Yuna Lee. She should be here today with us. So I really wanted to thank her for all the effort and expertise that she put for making these slides together. We work on several different projects in our lab, in particular for this portal level project where we create these atlases of single cell maps. The main two projects that I'll be talking about are the CMDKP and MSKKP, so knowledge portals for single cell, with the goal to create single cell maps for users on a variety of portals to make very complicated data intuitive for users to be able to scan for their favorite cell type, favorite gene, or favorite pathways in our portals. This is one example of one map that we have generated and uploaded to the portal. This is the biggest map that we have to date for pancreatic tissues, and we have, and actually, we are working on a manuscript. The person that did it in our lab is Weston Ellison. This is a huge map that is featuring data from 5 different sources, 327 subjects, and 5 different disease states across 3 different modalities with 3 different layers of cell type annotation. We have overall 15 tissues across these 2 different portals. For the CMDKP are mostly metabolism-related tissues like heart, liver, kidney, and so forth. And for MSKKP are mainly muscle-related tissue like skeletal muscle, intervertebral disc, or bone marrow. This is the workflow that we usually use and apply to all of our data sets. So the main step, the most complicated step, is to collect publicly available data from a variety of sources that I will describe later. And I will take this opportunity to ask the users, so if you have any single cell data sets or publication that, or like projects of publications that you would like to be features on our portal, it would be our greatest pleasure to feature them either as a reference if we believe that that is the best map that we can find out there, or as a query to be featured and integrated into our maps on the portal. So after we find the publicly available data sets, we scan all of them and we find the best possible single cell map that we can find. And we use that as a scaffold, as a backbone to project all other data sets on top of it to make every single map inherit different layers of cell type annotations. It goes without saying that all of this single data maps go through very rigorous quality control. And after we integrate them together, we make QC plots, create downstream assets like metadata and count matrices, peak set unified across our ecosystem, UMAP with different level of cell type annotation, differential expression, and accessibility if we have a TAC-seq or other modalities, cell communication assets, and enhancers, so Cris2Gene links. After we do all of that, we upload to Broad, and they do a great job making very intuitive visualizations for all these data sets. The resources that we use are publications. So like, this is one of the reasons why we do this, so that we can share the data and also like empower people to connect with us in case they would like to be features. PubMap, CellX Genes, CFD, like a number of sources across the net that we use to take all of the single cell data that we can find. This is an example of one data set that we're featured in our CMDKP portal, is the white adipose tissue or subcutaneous adipose tissue. If you go into AMP CMDKP single cell browser and you click on white adipose tissue, you will find a map like this. This features 700 donors and 1.5 million cells. And as you can see, there's like different layers of cell type annotations and expression, and you can look for, very intuitively, look for the expression of your favorite gene. Now I'll just go very briefly just to scan through all of the CMDKP and MSKKP maps that we have so far. This is like one summary slide to feature, for example, liver dataset. We have a number of, this is the breakdown by datasets. So in this case, we have 2, 4, 6, like 8 different publications. And the reference, this is a heat map for the number of donors. If we have information for controlling disease states, if we have information for age, sex, and then we can also like see the integration by dataset, by disease status, the different layers of cell type annotation, and the breakdown of the number of cell and the proportion of cell per dataset. This is the liver, this is the heart, this is the artery. All of our maps are like features hundreds of thousands of cells, if not millions of cells. Adipose visceral, skeletal muscle, adipose subcutaneous, hypothalamus. And for MSKKP, I will just show the tendon and the bone marrow. The other tissue are still in progress because the muscle community is a little, like the skeletal muscle tissues are a little bit more complicated to find data for, which is also one of the reasons why we're doing this. That will be the end of my presentation. If anyone has any question, please feel free to ask that. I'll be here to answer them. Okay.
Thank you,
Thank you for
Luca. That
having me.
was fantastic. I can start with a question while we're waiting for some other folks to trickle in. So I'm just curious. There are a lot of other single cell efforts that are out there in the community. How does what you're doing, compare or contrast to some of those other efforts? In terms of the scale, the types of tissues being used, the species being used, all of those types of things.
So as of now, we feature human and mouse. We have different models for different species, and the goal of this is to make a unified map by tissue unified map using the best reference that we can find. That is the main difference between the other type of efforts to do this. They are not focused on making tissue level maps. They just try to ramp all the data together in one big map that usually doesn't mean anything in that sense. So like I feel like the one of of our main um, pros that we have in comparison, for example, for human Atlas, is that we also ask the community, so you guys, to give us the best type of notation that are expertly curated to create tissue level maps that are outstanding in expertise and level.
Great, thank you.
Thank you, Luca. We have
Welcome.
a couple questions in the chat.
Yeah.
So the first is from Andy. Says, "Great talk. Do you have cell ontology IDs for the cell types in the bar graphs on your last slide? And how many unique cell types did you identify in liver, bone marrow, etc.?"
Okay, that's fantastic questions. Thank you, guys. Yes, we do have cell ontology ID. I believe we use Uberon ID. Ying is the person that does that in our lab, but incredibly grateful for her to do so because it's mainly manual scanning through Uberon to find the right Uberon ID for that exact cell type. So we do have that because there is an effort to have universal labels across the whole portal so that everyone can just look for a particular subtype of T cell and he or she will know that that will be exactly the same label across all of the data sets. For number of cells, that's a fantastic question. Like we have 3 layers of cell type annotation. And I like to cluster them in this way. We go by lineage, cell type, and subtype. Lineage is lymphoid, myeloid, hepatocytes, or so forth. So like big cluster of cells. Instead, cell type, you go to the cell type level. So for example, like lymphoid will be split into T cells and B cells. And then you go into the cell subtype level, and you can go T cell CD4, T cell CD8, and K cells, and so forth. The number of cell types, so like honestly, the number of lineage is kind of like 7 to 8 roughly across tissues because the lineage annotation level is pretty big, right? So that is roughly 7 to 8 across roughly any tissue level map that we have. For cell type and subtype, that is very much dependent on the tissue and on the reference that we chose to annotate those tissues from. Because sometimes papers go very crazy and they give us like 40 different types of cell subtypes, or sometimes it's just 20 or something like that. So it really depends on the map, but every map is available to the current version on the portals, and it should be very intuitive to go and look for any cell type that you are interested in.
Great. The
You're
next
welcome.
question is from Eva. And she's saying, we've been together for many years. Let's take a step back. I'm thinking big picture. So you might want to add the human being map of, a human body, with the pictures of the area first. This is awesome. And I think that's an interesting perspective of, how someone might want to enter into this type of mapping. So I don't know if we have plans for that or all, or what your thoughts are necessarily, but Eva, feel free to add on to what you had put in the comments.
Yeah, Eva, if you want to continue, you can do that.
Yeah, let's talk a little bit. Okay, so I worked
Yeah.
oncology and I'm a retired RN.
Mhm.
So basically what I'm after is part of what causes cancer is that we call it a mutation, right? But for instance, I tried to come up with an example and I'm on daylight savings time as you all are. And so a liver cell might suddenly start acting like, say, a pancreas cell.
Mhm.
And how is that? Well, it's putting out hormones that are wrong. And so those hormones are causing the immune system of the body to respond, and then it tries to turn them off, and then the next thing you know, we get a lot of inflammation and so forth. You put the pancreas on there, and we all know that part of what type 2 diabetes is the pancreas changes personality, and it becomes the more alpha cell dominant versus beta cell dominant. And so what I'm after, and it's very hard to write in the chat, but that as we advance, because we've all been together, I don't know how many years now, since before COVID, some of us on these webinars, that if we can identify, like picture a human body standing there, and you've got your cell types put on top of it, the in different colors, because we're lucky we can, with graphics, create a lot of colors. Then when we start thinking about cancer, we can change the color of the liver cell and make it look like a pancreas cell. And it's usually the part of the pancreas that does the digestive enzymes that causes, as you know, pancreatic cancer is a killer, and it's horrible. It's very painful, and it gets into the liver, and then it's a goner. People are a goner. We don't have a good treatment. So did I make sense? I mean, I
Yeah, I
used
get
way
it.
more words. It's because I needed another cup of coffee. I was teasing Mackenzie, but about 5 cups of coffee today. And then people are working on construction in the neighborhood where I am. So, oh, bless your heart, Luca. No, do you get me? We could
Yeah, I
do
get it, I
that
get it, I get it.
with
I get it, I get it, I
graphics.
get it.
Yeah, we
I
could
get it, I
do it
get
with
it.
graphics, right? Yeah,
Yeah,
we
yeah,
could
yeah.
do it
Like
with
I have an idea and
graphics.
I have an idea and an answer for that. So the short answer is that for now we decided to not take cancer into consideration exactly because of what you're talking about. Because like cancer behaves
Perfect.
like weird. And so like
Yes,
for the moment we decided to literally exclude cancer cells for this very phenomenon that you're describing.
perfect.
I would say I wouldn't exclude the possibility if the community is interested in like investigating also cancer cells in our maps. And something that we can do so like in the plans is to also on top of this create a map that is featuring all of these cell type annotations that are expertly curated from publications, but then take all of the tissues and put them all together in one big map. When we do
Perfect.
that, we will inherit the cell type annotations from the tissues, but we will cluster all the cells together. Actually, like we do have a better version of this that looks pretty okay already. And to your point, like what you would see when you do something like that. So like just imagine one interface where you have all of the tissues together. I don't know, like 10 million cells, right? And
Right,
then
right.
you go and look for your favorite cell type, and you could see that, for example, like cancer, like the sub. So you could, for example, just choose label cancer cells, and then you can go and see, I don't know, like pancreatic cells, cancer cells, and you can see that literally they move from one cluster of tissue to another because they
Perfect.
are still
Yeah,
labeled alphas or beta cells, but they are moving towards another cluster of tissues that is not the pancreas tissue. So you could
perfect. Yeah,
definitely see
that's
that.
it. No, that's it. And I don't even like working in oncology personally. That's why I am a diabetes specialist. But
Mhm.
it's the same concept, the alpha and beta, and then of course the other part of the pancreas is digestive enzymes, and we all ignore that. And some of those are really caustic. That
Yes.
they're damaging. So there's a lot of mucus cells that are involved in that. And it's been a long time since I took anatomy and physiology. So let's not, I don't know how long this webinar is supposed to be, and we don't want to get off on that, but you kind of get my idea. And
Yeah, I
so
do. I do.
I'm a woman that imagines things, not as they are, but as they could be. And that's why I
Yes.
keep coming to these webinars. So let me shut up, and I'm going to turn off the audio because I don't know how much more you had in planned for
Thank you very
this
much.
today. How much did we have planned for today? Do we have more?
So Eva, we wanted this to be an interactive discussion, so we're actually really glad that you're bringing this up. And I think that's our goal, right? Is to get the community together and, as you said, to start thinking about what we can do, what we've done already, and what we can do together to make more impactful resources. So thank you. I wanted to chime in. I think, Luca, your thoughts were fantastic on this. Eva, one thing that you mentioned that really stuck with me is your thoughts about could we have something visual, right, where we show the full human body and then show where a cell, lies on the map and all that. And I did just want to mention that we actually have a, partner resource that we work with through our Common Fund data ecosystem, HubMap, that's actually done a lot of that type of work and some really lovely visualizations in that space. They've developed things like a full body map, where you can see sort of slice by slice as you're going through and explore things using multimodal, sort of exploration tools, including some of those that exist in virtual reality. Andreas Buchel is actually here with us on the webinar, and he's one of the folks who's been heavily involved in that effort. And if he's interested, could speak more to that. But for now, I'll just put in the chat the link in case you're interested in taking a look at that as well.
Julie, since you called me out, I can say a few words. Hi, everyone. My name's Andy. I'm the research lead here in Kadi Bono's team at Indiana University. I'm going to put a link into the chat. This is our cell type populations paper that tries to, like Julie said, locate cells inside of 3D reference objects of the human body and inside of anatomical structures. This paper is accepted. We're currently in the proofing stage, and hopefully by the time we have our CFDE spring meeting in 2 weeks' time, it'll be published, hopefully. Let's see how fast the journal is. But yeah, this paper has all of the details alongside all of the figures that shows how this data was collected. HubMap is one single cell portal we used. We also used ZenNet and GTEx and Cell by Gene, but it's all in the paper. Thank you.
Thank you, Andy. That's fantastic.
Any other questions? Eva, go ahead.
I have a big mouth today. You caught me in a talking mood. So what he said brought another idea to mind, and I think what it is we might list out for our people that, we get a lot of people that see these webinars later, but they couldn't fit them into their schedule on the day that they happened. And so we might think about listing some of these maps and what they do in just an email. Right now, I'm getting this amazing email from somebody, some university about obesity, and they're listing all of the updates on obesity about every 2 weeks. We're also now have keto. I'm not going to push ketogenic metabolism on you today, but we have keto something or other, Keto Mojo on LinkedIn. Why that's interesting to us is even though a lot of us don't eat high fat, and I can send the genes out to some people that can really tolerate high fat versus some that do not. But we're all in ketogenic or alpha cell dominant metabolism when we sleep or when we do endurance exercise. And so this Keto Mojo thing has been really kind of awesome because it really is the pH of the cells that are causing them to mutate. That's why I brought up the thing about the pancreas, because I knew Luca would like to hear about they call it the acinar pancreas. Did I mispronounce the word? I have a speech impediment, so sometimes I screw up words, but basically, yeah, the acid base balance is partly why the people develop type 2 diabetes that's not reversible. Does that make sense? It's not reversible because they have too many alpha cells now to beta, and we can't push them the other direction no matter what we do with diet, exercise, and even the new GLP/GIP drugs do not correct it. Does that make sense? I haven't taught people in a while, and I'm working on graphics this week on teaching people what type 2 diabetes is. So that's partly what it is. Besides people being overweight and a lot of other problems we know that are associated with it, people stay in um, beta cell metabolism for maybe I'd say 18 out of 24 hours. And in our past, we were in beta cell metabolism for only 6 or 7 hours out of a 24-hour period. But that's partly my view on type 2 diabetes. So I'll shut up again and turn off the audio.
Well, thank you, Eva. That's an interesting perspective, and I hadn't heard, some of those thoughts. So I'm glad you shared that with us. Do you have any other, questions, comments for Luca? Thoughts of things that you would want to see in this resource to make it more useful to you?
We definitely would love to hear from the community. A lot of our visualizations and interactive tools be based off of things that, we think are useful, but it would be great to hear from the community members who might be using this in their own research. So please let us know, or if there's a specific even tissue that you're interested in seeing next. We're obviously working on different areas, but we'd like the community to be involved as much as possible with all of that. So we want to hear from you. Have members of the community, I'll ask maybe a question to everyone. Have people in the community used any of these tools before?
I'll say I have, but I think you know that already.
Yeah. I was just curious because I feel like if maybe people haven't seen it used before, we could give a quick showing of how people could walk through our resource and things like that. But
What
Andy?
would be helpful, Mackenzie, is what's the name of these tools? And so yeah, maybe a demo would be useful. Thank you.
Yeah, we can do that. Okay, so we have a single cell browser. I'm not sure which resource, Julie, do you think we should show it from?
We can do the CMDKP.
Okay.
And Luca, if you wanted to demo, I'm happy for you to do it. If not, I'm happy to do it as well.
I think it would be best if you do it. You
Okay,
have way
no
more
problem.
expertise. The
Mackenzie. Oh, no, you've done an amazing job on this resource. Here, give me just a second. I'll pull up
Yeah,
the link. Mackenzie, would you mind sharing that in the chat?
absolutely.
Thank you. All right. So this resource that Luca was talking about today, we have, basically the functionality of the single cell browsers available on a couple of our websites. One of the main ones that he mentioned today is our Common Metabolic Diseases Knowledge Portal, or CMDKP. You can get there by visiting cmdkp.org. And if you go to that page, you're here on our homepage. You can go over to KP Labs Single Cell Browser. And that will take you to a browser where you can get started, get sort of a bird's eye view of what tissues we have available at the moment to explore within the browser. And then you can see the interface is loading, and then we can start looking through it. So currently, we have selected artery as a tissue that we want to explore. This has, data from 14 human donors and 21 biosamples. I should mention on this page, these are all human maps at the moment. And Luca gave you some really nice context on how these maps were actually created. But now we can show you, how you can explore these interactively. So here you're looking at cell type clustering UMaps of, in this case, 11,000 cells from the artery data set. This is all interactive. So here you're coloring by cell type, and you can hover over any given cell type of interest to see that light up. But if you want to color by something else, so say, body mass index or sex, you can select those and change the way what is actually being showcased. If you go over here, you can see UMaps showing where your gene of interest is actually expressed on these cell type clusters. So here we've selected the MYH10 gene by default, but if you type another gene here, you can see that start loading up, and you can see which cluster that is expressed in. Sorry, my internet's a little off here. There we go. Okay, so you can see this is, predominantly in macrophages, but expressed at very low level, right? So then if you scroll down, you can explore these data in various, sort of aspects. So here we're looking at cell proportions by cell type within this data set. So you can see most cells in this particular data set are smooth muscle cells, followed by, the other cell types that are represented. And here you can see violin plots showing expression of your queried gene above, PPARG, in each cell type. So you can see, here where it's predominantly expressed. If you click these drop downs, you can get interactive visualizations with other categorical or continuous variables of interest. So here we're selecting body mass index, so you can see the cell proportions and the gene expression for that variable. If you scroll down further, you can see the top marker genes by cell type for each cell type within this data set. So that's sort of a basic overview of what the functionality is that we provide. So at the moment, we have this type of browser that we've developed for a couple of different resources. Today, we're focusing on the Common Metabolic Diseases Knowledge Portal, but as Luca said, we've made this available for a number of other resources, one of which is the Musculoskeletal Diseases Knowledge Portal, mskkp.org. As Luca said, these are currently available as maps for tendon and skeletal muscle tissue for human tissue. But Luca and Una are in the process of getting data available for additional cell and tissue types, as well as other species, right, Luca? Like mouse and yeah. So we're excited to build out that multi-species component here. And we've developed similar sort of resources through some of our other efforts. So I see some of the folks on the call from our PANC-based consortium. So I'll give that a shout out here. So if you're interested, particularly in the human pancreas and type 1 diabetes, we have a really nice resource available just for that through our integrated cell browser there. So you can check that out as well. Or if you're interested in, say, adipose tissue, you can go to our mammalian adipose tissue knowledge portal and go to our single cell browser there. So I'd say it's kind of a choose your own adventure situation where we have different resources that have adapted the same sort of core functionality and the same types of maps that Luca is describing for different, communities and applications. But that's the crux of what we have available. And you can explore some of our other analyses and visualizations on those sites as well.
Yeah, and just to echo, I know Noelle put this in the chat. We are looking for tissue champions. So if you are involved with, any of these analyses, or you have people in your organizations that you think would be a great fit for something like this, please let one of us know. Because, this is something that we're working towards building out better standards and also just a community as a whole. And we want, like I said earlier, we want to hear from you. We want to hear what tissues are important to you. We want to hear how to make the resource better, what you would like to add. So that's just one other pitch I'll say. And then we'll kind of pause for a second. Thank you, Julie, for showing that As I had mentioned before, we want to hear what you would like to see in these types of mapping. And if there's anything that you would like for the UI, we'd love to hear feedback on that as well. So I'll pause and do.
Yeah, and while we're waiting for, other comments to trickle in, I'll just add a little more flavor to the Tissue Champion initiative, since that's something we're really excited about. So this is an ongoing effort that, Luca, Mackenzie, Noelle, and I have been leading in collaboration with the folks from our musculoskeletal diseases knowledge portal. So the idea is that for each community, there exists, varying single cell RNA sequencing data sets, right? But what Luca and Una have actually found through their efforts is that the quality, utility of these data can vary dramatically from data set to data set. And Luca can give some more information about what I mean by that. But the crux of it is that if there are not clearly defined standards on what is required experimentally or computationally to make high quality RNA sequencing data, then the biological interpretation and integration of these maps becomes much more limited. So what we've been doing in this case is working with folks who are experts in either the experimental or computational side of RNA sequencing data generation and processing, and having them define for their tissue of expertise what the standards are and what they would recommend that others in the community follow in order to generate a good data set. So then we can improve the quality of data and integration into resources like these. And yeah, Luca, if you want to
Yeah,
chime in on that
yeah,
further.
exactly. Because like one of the things that we would like to implement, and I think this is something that in the Pancake portal we already have. I really like the idea of like tiers. So like if we can have like for Pancake it's easy because we have the experts on us. So we can like define tiers very easily across a number of PIs that are like top researchers in the field. And the idea is that we have a like gating system for a number of different data sets and a number of different types of data so that we can easily understand that, for example, like tier one is the best protocol that the tissue champion or like the expert of the field believe should be used to analyze that particular type of data. In this case, it's like single cell RNA-seq. So like tier one would be top quality data, top protocol for analyzing single cell RNA-seq for whatever tissue. Tier 2 is suboptimal type of data. Tier 3 is very particular niche experiment that needed to be done in a particular way. And so we know, so like if we had this very clearly defined protocol, and like it's like honestly, it's not even like how to analyze the data, because the end of the day, you just take the raw data and you reanalyze it again. So we can just unify across the board how we analyze the data, but it's more so what were the experimental conditions for this particular experiment to happen and what they could be to make it better. Because like one classic example is the like bone tissue is incredibly hard to come by and incredibly hard for obvious reasons to process. And so like we are like really looking forward for experts in the field to draw for us the dream experiment of like the top protocol that people should go for, so that we can take that into account when we make those maps and when we decide what is the best map that we can use for this particular tissue, so that we can just project everything in it. Does that make sense?
And I will add too, as we are building out this working group, we're also going to have sessions later in the year during this webinar series that'll be geared more towards the MSKKP and the Tissue Champions. So, as they share and come up with their standards, they'll be able to present that as well. So I just want to put that pitch out there to keep on your radar for future series as well. So. I guess I'll pause and see if anyone has any other questions, comments, thoughts about the UI. In addition to, I know we've talked a lot about the data, but if there's anything that people would like for the UI side as well. Okay. I'm not seeing any feedback on that. Okay, so I guess I'll just pause. Does anyone have any questions, concerns, any other items that you would like to ask Luca or our team? Eva?
Mute, sorry. I'm talking to myself. I got him off on that topic for a reason. So Luca, I might
Mhm.
send Mackenzie exactly what I'm talking about with my references. That way you guys can make sure. Yeah, there's so many layers because the cell type and the correct science is one piece. That's something I can't evaluate. I can evaluate the statistics and I can pull all the references of a particular paper and make sure that paper does indicate what, basically the paper itself has the correct references for the hypothesis. And so that's important for us because we need that. So we can look at levels because in order for the entire process at Broad Institute to be accurate, we absolutely do need to know if the science is correct, if the statistics are correct, and if the hypothesis really holds water. I mean, if it's good. So I tried to put the hypothesis in the chat, but let me r give Mackenzie the details and send her the references. Because you have the ability to look at any peer-reviewed paper, right? That I might refer to. Okay, because I've been at it 10 years and I've figured out what causes type 2 diabetes in quite a lot of detail. And so, yeah, let me have you guys look at it. You can shoot it down or grab it. Uh, as long as we know that it's like a peer-reviewed paper that hasn't been published, it needs to be confidential. But yeah, I put part of it in there and I hope that helps you. I figured part of it out and it's so complicated because the balance of alpha and beta cells has been knocked out over the last 200 years and we can't we can't go back 200 years ago and see what the cells look like. There is no way to do it. The only potential place was an Iceman, somebody that was found frozen in the Alps. They were able to dissect his body and it had not been, nobody used formalin on it yet. If we find another body like that, we might we might be able to look at the alpha beta cell complement and figure it out. But so yeah. So I will send it all to Mackenzie. Now, I didn't even hear the beginning because I was off trying to answer my front door. But what date is are you going to the American Diabetes, right? The
Yes,
big
we will
thing.
be there. Hmm.
What date was that, Mackenzie? Remind me.
Yes, so it's in June, and it is the 5th through 8th.
And it's in New Orleans this year, right?
Yes, it is.
Yeah, so I can drive there. It'll all be good. It'll all work out.
Awesome.
But yeah, for the last 2 weeks, I've been putting all of my 10 years of research into different organizational chapters. But part of it is the science, and I can only evaluate the science by looking at the statistics and looking at who else got this result at another lab. I can't I can't do the science. I mean, I have a master's in public health, but I was taught to evaluate papers quite intensely. So you get me, right? And so, yeah. So thank you. You guys,
Awesome.
this has been awesome. And so let me write it up and send it to Mackenzie, and then she'll have my references.
That sounds great. Thanks,
And
Eva.
you can share them with Luca. I think I trust him enough. Thank you.
Sounds
Thank you
great.
very
Thank
much.
you so much. Okay, Andy?
Luca, I have one more question for you. And you might
Yeah,
have covered this and I might have missed it.
no
How did
problem.
you decide which data sets to use for your maps? Like, I assume you probably had some kind of quality criteria. I'm curious about, the process you had in place to select, like, include and exclude data sets.
Yes. So like other than looking, so we have a few standards that we really like to have. First of all, the better, more updated type of experiments we tend to prefer. For example, if we have single cell RNA-seq or single nuclear RNA-seq, we would like to go to single nuclear RNA-seq. That's usually like newer than single cell. Multiome would be even better because that has higher resolution and we can make use of the ATAC-seq datasets. That is like the first thing. So like there is first check is like what type of assay these people did first. Second, if it's from published datasets, like the publication level, it's also part of the reason why we choose a particular dataset. If we have a science paper or if we have a like science advances paper and the science paper, like the data looks better, we'll go for the science paper. So like we go for like publication quality. Then we go and look for QC data. So like there are very, very like easy to see distributions of features that we go and look by like barcode. For example, like good data set tend to have a very low percentage of mitochondrial genes and very high number of genes per cell. So if a data set doesn't have the correct distribution of features, we tend to either not even use the data set, or if we like the data set for whatever reason, maybe there's a lot of donors or there's a lot of cells, then we tend to use it as a query instead of as a reference. And then we clean the query using the reference mapping scores. Because when you do reference mapping, you're projecting labels from one data set to another, and then you have a score that tells you how confident that projection was. And so you can just clean cells that were not confidently being projected over the actual sets that you believe in. And other than that, as I said, we look for subjects, like how many people were there, how full is the metadata, were there disease states or not, how many cells, and all of that stuff. Like it is a like pretty integrated approach to figure out what is the best data set. Does it
Excellent.
make sense?
Thank you. Yeah,
Cool.
that makes a
Yeah.
lot of sense. Because that preprint I was putting into the chat earlier, we have similar issues where we need to decide which data do we include and which data do we not include. So I'd love to
Yeah,
have
yeah.
a more exchange about that. Are you going to the CFDE meeting in 2 weeks?
Probably not, but you can reach out to Mackenzie and Julie, and they can give you my like email if you want to chat more. No problem.
I have your email. I'll send you an
Oh
email.
yeah, there you go. Yeah, yeah. Just send me an email, and we can Zoom anytime. And if you want to collaborate, we can collaborate too. No problem.
Excellent. Thank you.
Yeah, yeah. You're welcome.
Great. Thank you all for this discussion. All right, we have a few minutes left. So I just want to see if there are any other final thoughts, comments, questions for the team. And one other thing I will say, if you are using our site and you have any questions or concerns, we would be happy to meet with you to discuss if there's anything that you want personal in-depth help with. You know, sometimes some of this, it's just easy to talk one-on-one instead. So I just want to offer that as well. Any other questions? Okay, I am taking silence as no more questions. So with that, I think we can wrap up a little bit early. Thank you so much, Luca, for taking the time today to go over the single cell mapping. I think this is going to be a great topic that we continue on as we go. So again, if anyone has anything that comes up between now and then, we can always talk about this again at another webinar. And with that, I just want to give one more reminder that we are going to be updating the webinar series, so it's going to be bi-weekly. So if you got emails from me, I'm sorry, there might be a few with the updated webinar structure. So thank you again. This is going to be up on our YouTube and news items as well. So if you want to share with any colleagues that were unable to join, we'd be happy to do that as well. So big thank you again to Luca, and we really appreciate you joining us today. Thanks everyone so much for the active discussion, and have a great rest of your day.
Thank you.
Thanks, everyone. Bye.
Thank you so much.
Bye.